Thank you for attending this, everybody. Your lovely evening. I don't know if it's so lovely there, but you can see the sun is shining on my face. It's a very nice evening here. So thank you for spending it with me. So just to introduce myself a little bit, I'm a postdoc at the University of British Columbia and also cross-appointed at North Carolina State University. So I work very closely with David Tarpey, and also Jeff Pettis is one of my co-advisors over there, along with Leonard Foster at UBC. So I may be cross-appointed, but I spend most of my time, especially during the pandemic, here in Vancouver, which is where I am right now. And I, over the last few years, have been working fairly closely with some beekeepers here in BC to do these surveys of queens, which I will talk about. It's the meatiest part of my talk, so I'm pretty excited to share that data with you. So I'll be going through a few different questions in this talk. First is, why study honeybee sperm and temperature stress in the first place? It's not necessarily an obvious issue that we should be looking at. So hopefully by the end of this, I can convince you that, that it is worth investigating. And what is the Goldilocks zone? So what is the safe temperature range that queens should be kept in at all times to minimize damage to their fertility? And also, of course, what can we learn from surveying failed and healthy queens from actual beekeeping operations? So, sorry, I need to get rid of this, this bar at the top of my screen because I can't see the things I have written. So why study honeybee sperm and temperature stress in the first place? Well, the thing that really got me interested in this project was just that it is such a biologically interesting system of reproduction. So queens, as you probably know, have a spermatheca, which is where they store all the sperm that they'll need to fertilize eggs for their lifetime. And actually, almost all insects have a spermatheca of some kind. And even some amphibians and reptiles, if you believe it. But in honeybees, it's particularly well developed. So it's this very neat organ. It just looks like a little pearl towards the back of her abdomen. And when the queen mates early in life, she'll acquire millions of sperm from dozens of drones. And actually, when she first mates, there are hundreds of millions of sperm that enter her reproductive tract, and they're sort of held in the oviduct area for a little while. And then a small fraction of those will migrate into the spermatheca, where they will remain for hopefully years. If you're lucky, your queen will live a few years, but these days it's often much less than that. So every time the queen lays an egg, let's say an egg I lost my cursor, sorry, there it is. Every time an egg comes down the oviduct, it'll sort of press against this little S-curve thing. And then if she's choosing to fertilize it, then she'll sort of squeeze this sperm pump, and that releases a teeny tiny amount of fluid from inside the spermatheca to fertilize that egg, I should say, containing sperm to fertilize that egg. And on average, there are about 2 sperm in that small volume, 2.5 picoliters, or millionths of a milliliter, that she releases per egg. I like to think of it as like 1 sperm cell to fertilize the egg and then another one for good luck. And so that means that her lifetime reproductive capacity is ultimately limited by how many viable sperm she has. And in the best case scenario, a queen, like a well-mated queen, will have a lifetime fecundity of about 1.5 million eggs. And that's a lot of eggs. That's like 6 years worth of eggs. But as you can imagine, queens are not always well mated. And even if they are, there are all kinds of things that can happen to them to kill the sperm that she has stored, bringing her reproductive capacity pretty far down. So previous research has shown that queen failure is linked to low sperm viability. This was work done by Jeff and published a few years ago now. And temperature stress has actually been proposed as a potential causal factor. And that may not be all that surprising because temperature stress actually kills sperm in all kinds of different animals, including people. So it's not surprising that temperature stress would kill the sperm stored within queens as well. And what about pesticides? Well, I guess I don't really have to go into this anymore because I sort of explained in the answer to Jim's question that all the research I've done so far on queen exposure to pesticides has shown that it doesn't really have a direct impact on her quality. So I will be talking mainly about temperature stress today. So I should say also there, in summary of all those points, it's both a very biologically interesting system to look into, but there's also an economic aspect because queen failure is reported so often by beekeepers these days that any knowledge we can generate about why that might be happening or what biological processes enable the queen to sustain sperm viability in the first place may be useful. So you might wonder though, well, okay, temperature stress, maybe it doesn't really matter for honeybees because the queen spends most of her time inside the hive. You know, she's only really out exposed to ambient temperatures when she's mating or when she's swarming. So why do we care about temperature stress? Well, humans really like to stuff queens in little tiny cages and then ship them halfway around the world with just a few attendant workers in those cages. And in that kind of a scenario, they have a hard time thermoregulating. So there have been a few studies now, including one that I published last year, where we've tracked the temperature variation in just regular queen shipments. And here's an example of some of that data that we published. So here you've got temperature on the y-axis and then just the duration of the shipment on the x-axis, and each squiggly line is just showing how the temperature changed over time as the queens were being shipped to our lab. And most of these were ground shipments, and one of them came actually from Edmonton to Vancouver by air transportation. And that one actually was the one that had this really low spike down to 4 degrees. It did not occur during the flight. It occurred based on the timestamp. It was exactly when the queens were sitting in the Air Canada warehouse waiting for customs to clear them. This was in the middle of June, and I still can't figure out how they possibly got this cold because it was not that cold outside even. So it's like the only thing I can think of is if somebody saw the do not expose to sunlight sign and thought it was a good idea to instead put them in the fridge or something. I don't know, but it got quite cold. And some other people have found quite a bit of temperature variation as well, though not as extreme as we found in these 8 shipments. But if you sort of look at everybody's data together, we see what I call an extreme temperature, and I'll get into what exactly that is in a little bit, in 10% approximately. About 10% of the shipments have an extreme temperature event. And, you know, this could potentially be a pretty big problem, especially in Canada, because we import around 250,000 queens every year from other countries. So that's a lot of queens that are being shipped. And if 10% of shipments have an extreme temperature event, then that could potentially be a lot of damaged queens out there. So I also want to push back a little bit about this idea that colonies are these super thermoregulated environments and that we don't really have to worry about what goes on inside them. There have been some reports of temperature spikes greater than 40 degrees that I could find in the literature. And I wanted to know also what happened inside colonies during really extreme weather events. So like during a heat wave, are the bees still able to keep the temperature cool enough inside, for example? So this again was work done in collaboration with Jeff. And here temperature loggers were placed on, they were stuck on each frame of a 10 frame deep wooden colony. So just a single box, but 10 frames. And the darker colors are the temperatures of the frames on the outside, and then the lighter colors are the ones that are like in the core. And the blue line is just the ambient temperature from a temperature logger that was placed in the shade underneath each hive. So this was done in California, not in BC, where they had this really bad heat wave. I don't actually remember exactly when this was, but the temperatures got up to about 45 degrees outside, which is pretty extreme. And what you can see in this data is that the core, as you might expect, the core is maintaining a pretty stable 35 or 36 degrees. But the farther that you get towards the edge, then the more variation in temperature you have. So in this hive, Hive 2, you can see that 3 frames in from the outside, you're still reaching temperatures of around 39 degrees, which is pretty hot. So, you know, this data is not saying that these queens were exposed to temperature stress during this heat wave, because they very well could just be wandering around in the core, which is probably where you would expect them to be. But, you know, this is a 10 frame colony, and sometimes we have 5 frame nukes, or we have 3 frame mating nukes, or mini nukes for mating queens in. And I haven't collected data from smaller colonies, but if I were to guess, I would say that they are probably less able to thermoregulate than these big ones. You know, if you think of, so this hive 2, 3 frames in from the outside is getting to be 39 degrees. Even in a 5 frame colony, 3 frames in from the outside is like the middle frame. So, you know, there's a question of, well, maybe it's easier in the smaller space, but if I were to guess, I'd say it's probably more difficult for the smaller colonies to thermoregulate than the bigger colonies. So that is showing that there's a lot of potential for extreme temperature exposure, both during shipping, where normally the extreme temperatures are more on the cold end, but also potentially inside hives. But it doesn't tell us like how hot is too hot and how cold is too cold. So we did another pretty big experiment where we took almost 200 queens and just exposed them to a whole range of different temperatures, from 5 degrees all the way up to 42 degrees, and for 3 different durations: 1 hour, 2 hours, or 4 hours of temperature stress. And then after giving them a couple days to kind of recover, then they were sacrificed, and we measured the sperm viability using this technique called fluorescent microscopy, which is where we just stain the sperm different colors. We can stain them red if they're dead and green if they're live, and then we can just count up how many dead ones and how many red ones and how many green ones there are to figure out the sperm viability. So when we did this, now keep in mind there are like a lot of queens here, and I'm not showing each individual queen's data. I'm just showing you the average across the temperature range for clarity. But after 1 hour, you can see the sperm viability is not really changing. It's there's a little tiny bit of a bump here around 30 degrees, but there weren't any significant differences across the temperature range for that duration. But for 2 hours or more, and I've pooled the 2 and 4 hour data here into one graph because they were very similar. Here you see a pretty clear trend where there's low viability at the cold end. You see this drop here, and low viability up here at the hot end as well. And this pretty clear bump in the middle. And so that still doesn't totally answer the question of how hot is too hot and how cold is too cold, because we need to know, sorry, I keep losing my cursor, it's super annoying. We need to know like how far down this line we have to go before it's too much loss of viability, essentially. So I defined that tolerance threshold as 11.5%, for reasons that you'll see in a couple slides. But that was the number that I think best represents the threshold for like, okay, if you lose this much sperm viability, then we know that that's a bad thing. So if I set that as the tolerance threshold and then essentially look at where an 11.5% drop intersects with this average line, then that tells me that keeping the queens between 15 degrees Celsius and 38 degrees Celsius will keep you below that loss of sperm viability. So this, in summary, like the 15 to 38 degrees is like the recommended temperature range that I think queens should be kept at. You know, ideally, even here, I need to change this cursor thing. It just keeps disappearing. Okay, there we go. Ideally, you would want to keep them more towards this bump. So probably between 25 and 35 would be ideal, but really, really try for at least 15 to 38. So to summarize this section, I hope that now I've shown you that queens are vulnerable to temperature stress, particularly cold temperatures during shipping, and in small colonies, queens could be vulnerable to extreme temperatures as well. And that 15 to 38 degrees is the recommended temperature range that queens should remain in at all times, especially during shipping. So on to the final part, which is what can we learn from surveying failed and healthy queens from beekeepers? So this project actually started because some beekeepers here in BC asked me to compare the quality of their local queens in terms of sperm viability and sperm counts to imported queens from California and Hawaii. So this was a really interesting question, and I said, yes, that sounds good. And while you're at it, maybe you could send me also any failed queens that you have in your operation and see what we can learn from this really big sample. So I don't know if you're familiar with the term scope creep, but in science, so often we start with a really fairly simple project with a straightforward question, and then we just get more and more ideas as the project develops over time. So this simple request from some beekeepers in the BC BBA turned into us measuring not only sperm viability and sperm counts, but also their viral abundances and their ovary mass. And I thought, oh, what the heck, maybe I'll just do some protein measurements on their spermathecal fluid while I'm at it. And actually now another student in the lab has also started looking at proteins expressed in their ovaries as well as in their hemolymph. So we have this really big data set on all these queens now, and I think it's safe to say that they have thoroughly donated their bodies to science. So I'll share with you just a little bit of what all this data is telling us, starting with the sperm viability and sperm counts. So I'm not actually showing the imported queens here, but the answer to the original question of how do the local queens and imported queens compare was that they're virtually identical. They were indistinguishable in terms of their sperm metrics. The imported queens did have significantly smaller ovaries, but I'm pretty sure that that was just an effect of them being caged for longer than these local queens because they had to be shipped a greater distance to get to me. And when I put those imported queens into a queen bank and then measured ovaries of them a couple weeks after, those ones had bounced back. So I think that was not really an honest indicator of them being lower quality. It's just that the ovary size is it's like a very plastic kind of measurement. So they were virtually identical. And the failed queens, though, compared to the healthy queens, they had much more variable sperm viability, as well as more variable counts and more variable ovary mass. So here, the size of the circle is proportional to the size of the ovaries. And there's an eensy weensy little ovary queen here, and then this giant one over here. And there's a bigger spread in the viability and in the number of sperm that they contain, probably because I was including queens from colonies with all kinds of symptoms, not just symptoms that I might suspect were linked to their sperm metrics. So I think that's contributing to the variation. But even with that, there was still a significant drop in sperm viability. And this is where that 11.5% number comes from. So 11.5% is the viability difference, the sperm viability difference between failed and healthy queens. So that is why I used that as the tolerance threshold in that temperature Kirby graph, because this is the loss beyond which I think it's pretty safe to say that it's biologically and economically an issue. Sorry, there are lots of like little windows blocking things. Sorry, it's like Zoom management issues. So failed queens have lower sperm viability, and even when I'm including this really diverse sample of queens, but this wasn't really a perfect experiment because the failed queens often had unknown ages, and I think it's pretty safe to assume that they were biased towards being older than the healthy queens. So I really needed to figure out if these metrics were also linked to the queen's age, to know just how useful these differences really are. So the next year, I essentially repeated this survey, this time using queens just from the Lower Mainland and queens where I knew how old they were. So here we have queens from 2018, 19, and 2020, and we have old healthy queens as well as young failed queens represented in this mix. So even when I account for queen age, there is still a significant drop in sperm viability in the failed queens. So this is a very reproducible result. And the drop was a little bit higher, 12.3 compared to 1.5%, but I think, overall that's still pretty similar. And I think 11.5% is still the better number to use for figuring out that the temperature range to keep queens in, because if we're making these recommendations to beekeepers, we kind of want to err on the side of caution. So I think using the smaller number is probably safer. So that's what's going on with the sperm, but now what's happening with the viruses and the ovaries? And these two things actually appear to be quite closely linked. So this graph is a little bit weird if you're not used to looking at this style of graph. So first I'll just explain. This on the y-axis is the viral copies. So you can think of that as like how many viruses are there. And we measured 3 different viruses: deformed wing virus, sac-brood virus, and black queen cell virus, which are all known to infect queens. And the black dot represents the median. So that means that that's the number where half the samples fell below that number, or at or below that number, and half are at or above. And the width of this kind of violin shape is proportional to like how many data points there are at that number. So this really wide sac-brood virus plot just means that a lot of the queens had 0 detectable sac-brood. So hopefully that makes sense. But if it doesn't, then maybe take my word for it that this is showing that the failed queens had significantly lower levels of DWV, which was kind of surprising, but they had higher levels of sac-brood and higher levels of black queen cell virus. And if you actually add all these different viruses into one like total viral load factor, then the failed queens have higher viral loads overall, kind of like regardless of which type of virus it was. So failed queens are sicker than healthy queens. Maybe that's not surprising at all. But not very many people have actually measured this, and the queens are not, they're not like overtly sick. They don't have any visible symptoms of viruses. And actually, these levels of viruses are very, very low. So they're totally asymptomatic, but it might still be having an impact on them. So when we looked at the relationship between the total viral load and the size of the queen's ovaries, we found that the queens with more viruses had smaller ovaries. So there was a significant negative correlation between viral loads and ovary size for both failed queens and healthy queens. So the healthy queens, of course, had lower viral loads overall, but there was still the same negative trend there. So this is, I think, really worth investigating further to see if there's actually like a causal relationship. So we're planning to do experiments this summer where we infect queens with viruses and see if their ovaries are any different, all else being equal, because this was a big study, but it's all correlative. So we can only speculate at there being a causal relationship here. But it's definitely worth looking into more, because this relationship between ovary size and the queen's quality is pretty consistent. So in all the surveys that I've done so far, using queens from many different places, this was from South Central BC, these were queens from Pennsylvania, and these were queens from the Lower Mainland, we see the same trend that failed queens have smaller ovaries. So this, I think, is an important factor to look at. And we don't have virus data for all these queens yet, but I'm really curious to see if the same relationship holds. So what we think is going on is something like the queens, when they have these low-level infections, they're spending too many resources on trying to clear that infection or their immune response against those viruses, that they can't invest as many resources into egg production. That's sort of the going hypothesis that we're going to test. So now what about the proteomics? This is the last bit. Maybe I saved the best for last, I think. I don't know, depends on what you like, I guess. But one of the things I was super interested in going into this was just figuring out what proteins in the spermathecal fluid are related to sperm viability, because I really just have a burning curiosity to really figure out how queens are able to keep sperm alive for so long. I think it's a really amazing ability. And so one of the first things that I wanted to look at was just what proteins are correlated with sperm viability. Like not even, not looking at failed versus healthy queens, just looking at the natural variation in sperm viability, what proteins are related to that. So I found a handful of proteins that are significantly correlated, but I'm only going to bring your attention to one of them here. That's lysozyme. And lysozyme is actually a very conserved innate immune protein, normally used to fight off bacterial infections. And so here we have kind of like another version of the same story, where the queens that have higher amounts of lysozyme have lower sperm viability. So it's kind of all pointing into the same direction, that when queens are launching or are having to launch an immune response, that their fertility suffers. So whether it's sperm viability or ovary size, as we were seeing before with the viruses. So that's pretty interesting. I think we really need to do more research on queen pathogens and effects of pathogens on their physiology. Speaking of pathogens, we have the viral data for all these queens. So the next thing I did was to look to see what spermathecal proteins are related to viral abundance. And again, there were not very many, but there were some that were significantly correlated. Here we have the protein amount on the y-axis, like before, and then the viral load, total viral load on the x-axis. And I just want to talk about these top proteins here because they are actually all of this group of proteins called heat shock proteins. And that, okay, we're kind of coming full circle now back to temperature and heat stuff. That was initially quite confusing, but luckily for me, last year, some researchers at Montana State University published a really great article on the effects of heat shock on workers' ability to clear viruses. So they actually found that this heat shock, the heat shock response, so upregulating heat shock proteins, actually has an antiviral effect. So in their experiment, they had workers and they infected them with this virus. It's not actually a normal honeybee virus, but it does infect honeybee cells. So they infected them with this virus, and then half of the workers, they then heat shocked them, and the other half were not heat shocked. And the workers that were heat shocked were significantly better at clearing that viral infection than the workers that were not heat shocked. So they sort of talk about these heat shock proteins as being kind of like oil to the machinery, the regular machinery that helps them fight off virus infections. And this this is kind of problematic because one of my other experiments is to try to see if I can find proteins in the queen spermathecae that indicate different types of stress. And of course, you know, working on temperature, one of those things was heat stress. So I did all these experiments. I stressed queens and measured the proteins, and the ones that were looking like the best indicators of heat stress were actually exactly these proteins. So that means that my grand plan of being able to take a failed queen from a beekeeper and run these lab tests on her to try to figure out what she might have been exposed to cause failure will totally not work, because two of my markers actually completely overlap with the proteins that are also upregulated from viral infection. So that's too bad, but it's also there's also kind of a way forward here. And this data, it's really strongly suggesting that the heat shock response is antiviral in queens as well. And so something that I plan to do this summer is to see if heat shocking adult virgin queens, so before they've acquired sperm, so I'm not going to be killing their sperm by heat shocking them, if heat shocking them can help protect them or prime their immune system to better defend against viruses that are transmitted to them during mating. So that is a known transmission route for viruses, that they can be passed from drones to queens during mating. And it's possible that this might be kind of a cheap and easy way to give your queens a fighting chance. But that, you know, that's yet to be tested. That's going to be what's keeping me busy this summer. So with that, if the rest of this talk was a blur to you, then just remember these key points. And that is that temperature stress kills honeybee sperm. And it can happen inside hives as well as during shipments. And I should qualify that with inside hives it would probably only be a risk for very small colonies and during very extreme weather events. What is the Goldilocks zone? Well, right now I'm recommending to keep queens between 15 and 38 degrees. If you can get closer, keep it closer to that 25 to 35 range, then that's even better. But these are sort of the lower and upper limits that that I would be willing to take anyway. And what can we learn from all these queen samples that we've acquired? Well, the failed queens have fewer sperm, lower sperm viability, higher viral loads, and smaller ovaries. And also high levels of this conserved immune protein is linked to low sperm viability. So we're getting the same story from a couple different angles here. And finally, that it looks like heat shock proteins are an antiviral response in queens as well. And hopefully there's a way that we can exploit that. So With that, I'll just say thank you to everybody who's helped me throughout this. I've had so much help from so many different people, particularly beekeepers as well. So big shout out to them. And also, of course, the funding agencies who have all helped to pay for this work. And to you for listening to me.